Well, something's heating up. What could that mean? Well, it could be something to do with autoclaves. Well, kind of. We're going to have a look at biological indicators, which are used to verify steam sterilization. Indeed I do. Well, hello, Tim Sandle back with you with another video, and this week's subject is biological indicators, which are used to verify whether a sterilization or decontamination system is working effectively. And in this context, we're going to be looking at biological indicators that are used for the verification of moist heat or steam sterilization, as you would with an autoclave. So on the screen we have a fairly standard autoclave used in pharmaceutical facilities with sort of standard digital displays, and this is like a gravity displacement autoclave, as you may well come across. Okay, so to verify that an autoclave is working effectively, we need to use a biological indicator, and this is part of the biovalidation process. So we would operate our autoclave as part of the validation, and we would map it for heat distribution using thermocouples, and then we use some of those locations, also take into account the mass and surface area of the load, and look for more challenging locations where we may not get steam penetration, and use that to position a number of biological indicators, typically 20 for a standard autoclave cycle. Our biological indicators are generally presented as spore strips, which are strips of a little bit something resembling filter paper, which contain bacterial endospores, or they're in ampules which are designed to capture fluid loads as well. So there's some key characteristics that make for a good biological indicator. The first thing is that we want our biological indicator to be pure. So we want it to contain the spores of the microorganism that we're most interested in. So for steam sterilization, we have a bacterium called Geobacillus stereothermophilus, and this organism is resistant to moist heat, and this kind of relates to its origins. So this particular organism is held in culture collections around the world, and the origin of it is from the hot springs or geysers that erupt from the ground, and this particular strain was isolated in Yellowstone Park in the USA. So it's used to surviving in temperatures in the 90s bordering on 100 degrees Celsius, so it makes it a suitable organism for verifying autoclaves. And here we just have an illustration. This is where somebody called Kerry Skinner has attempted to populate an item of rubber, in this case a rubber glove, with bacterial endospores, and you can, if you look like to the left hand side of the photograph, you can see distributions of spores in the undulating folds of the rubber. Okay, another important factor is the population. So the minimum sterility assurance level, which is the verification or the confidence level of non-sterility of one item surviving, is 10 to the 6. So we're trying to seek a million bacterial cell reduction. So we want to have biological indicators that have a million or more cells on them. So we need to have the right population, and there's certain standards in place about verifying that population, which should be something either done by the receiving laboratory or based on independent testing or taking manufacturer's certification. But we want to have about 1 million spores on the biological indicator. Okay, but perhaps the most important characteristic of the biological indicator is this thing called the D value. So D is decimal reduction value. And this is an indication of the level of resistance. So the D value is assessed against a temperature. And the D value is the time taken to reduce the population of microorganisms by 1 log or 90%. So for example, if we're running an autoclave at 121 degrees and we purchase biological indicators with a D value for 121 degrees Celsius, it might be quoted as 1.5 or a value higher up till 3. So if it comes as 1.5, that means it takes 1.5 minutes to reduce that population by 1 log. And therefore, if it's for 3 minutes, double the time, we'd have reduced it by 2 logs and so on and so forth. So we can use that to assess how long do we need to run the autoclave for in order to get the 6 log reduction of the population of the spore challenge. So the biological indicators are purchased against criteria we set out in the European or United States Pharmacopoeia. And so we have to have these biological indicators with a D value of 1.5 to 3 minutes. And we put about, as I said before, about 20 of these into a typical autoclave, looking for worst case spots or areas such as inside tubing where we may not get adequate steam penetration. Now, the way that biological indicators are assessed, which is done by the manufacturer using specialist equipment, is a device called a beer vessel. Unfortunately, that's not beer to drink, excuse me a second, but it's beer, B-I-E-R, which stands for biological indicator resistometer. And it's a special device for assessing D values. It's quite dangerous to operate because it's like running an autoclave, stopping it, taking out a tube, running it again, stopping it, taking out a tube, running it again, stopping it, taking out a tube, running it again, stopping it, taking out a tube, running it again, stopping it, taking out a tube, running it again, stopping it, taking out a tube, running it again, stopping out a tube, and you kind of get the picture. You're trying to work out the points where you're getting survivors and then you're getting killed. So that's the way to assess how well a biological indicator is working and what the D value, calculated D value is, which is kind of subject to a calculation. We also want biological indicators to be assessed for their shelf life, and we also sometimes want to know other factors, things like Z values, which you don't need to worry yourselves about. But if you want to run an autoclave at a hotter temperature, like 134 degrees, you want to know, can you reduce the time by having a higher temperature? Yes, you can. How do you go about that? Well, there's a series of mathematical calculations, and things like Z values help with that assessment. Okay, that brings this particular video to an end. As usual, thank you for watching. I'm Tim Sandle, and I'll be back with you before you know it. Cheerio, bye-bye.